Welcome back. Good morning, good afternoon, good evening, whatever time it is when you're watching this. I just experienced some chihuahua noise in the background as well as some kind of argument. So hopefully the video won't be too interrupted by the surroundings here. I'm going to try to keep this one similar length as last time and wrap up the lecture on Homo Sapiens. So let's go back just to review some of the things that we already talked about in class. The Australopith species, like the species in the genus Australopithecus, there were many different species, right? But that genus, the species of that genus, were around from about 4.5 million years ago to around 1 million years ago. So you see on that lovely timeline there. And that's a reconstruction of perhaps what one of them, like, for example, Lucy might have looked like. And we know there were gracile Australopiths and robust Australopiths. And then after that, there was Homo habilis, which was one of the first, if not the first, Homo species. And Homo habilis was around from about 2.5 million years ago to 1.8 million years ago, right? And Homo habilis is probably the first species to be associated with stone tools directly. And then Homo erectus was actually the first hominin species to migrate beyond the continent of Africa. So Homo erectus was throughout large parts of the Old World, Asia, Europe, and even down into Southeast Asia, probably on some of the islands. We think maybe a population of Homo erectus was on the island of Flores in Indonesia, and through a process of island dwarfism, eventually sort of became Homo floresiensis. But anyway, Homo erectus was definitely had a larger cranial capacity for the first time, like a significantly larger one, and had a cultural toolkit probably, and a literal toolkit, a Schulian technology that enabled it to travel far and wide. Then we have Homo heidelbergensis in both Africa and Europe. Came on the scene somewhere between 1 million and 500,000 years ago, and was no longer by 300,000 years ago. But Homo heidelbergensis, probably populations of heidelbergensis, sort of through a process of what we call anagenesis, like kind of evolved into what is now anatomically modern Homo sapiens. So our species is thought to be around 200,000 years old, approximately. Could be a little bit older than that. Some things have come to light recently, which might push that back a little bit. Now, as far as, like, again, the start time of Neanderthals, I'm not going to really be adamant about this particular start time, because I think that a lot of sources will put it quite a bit earlier than this. But at any rate, Neanderthals were around until about 30,000 years ago. And that's pretty secure. Okay, so that's our timeline. So what are some of the characteristics in terms of skeletal morphology of AM is anatomically modern Homo sapiens? We have a more orthognathic face, which means a flatter face. It doesn't come out quite as much as the earlier species. So think of orthodontist, straightening your teeth. So orthognathic means flatter. More of a vertical forehead. See, I'm not wearing my hat today. And I come to you with less hair than yesterday, so I can model appropriately. The back of the head is more rounded. We have a chin, and again, I don't know how many times I've used this joke now, but I know I don't have like a Henry Cavill style chin, but there is one there, right? It comes out a bit. And the earlier species didn't have it, so we're the only hominid species that actually have a chin. It's puzzling why, we don't really know. The skull is more rounded in the back, as I said. We don't have that occipital bun like the Neanderthals did. And our cranial capacity, on average, is around 1,400 cubic centimeters. Okay. Our skeleton is actually less massive than that of the earlier species, especially like Neanderthals. That's not to say it's super fragile, but compared to the others, it's slimmer, it's lighter. Probably the bone isn't as dense. The muscle attachment areas are not as sort of projecting as, say, with Neanderthals. And some of that just has to do with changes in lifestyle as well, and climate. So here's a comparison on the top of a Neanderthal skull. You can see the face kind of projects out a little bit more. The skull is longer, the forehead is lower, and there's not really any chin. And then we have a more of a like an early anatomically modern Homo sapiens skull there on the bottom with a flatter face, a bit less massive supraorbital torus / brow ridge, and no occipital bun. Okay, already said all that. All right, so we think that this transition between Heidelbergensis and what we now consider anatomically modern Homo sapiens occurred somewhere between 300,000 years ago and 200,000 years ago in Africa. We have found some fossils that appear to be sort of transitional, that have characteristics that are similar to Heidelbergensis, but also approaching what we would call anatomically modern Homo sapiens. One of those was found in Morocco around 315,000 years, was 315,000 years old, and that's the skull on the bottom, and the picture on the top is like an artist's rendition of what the person who that skull belonged to might have looked like. There's another, probably like one of the earliest, more certain Homo sapiens, anatomically modern Homo sapiens skulls, is also from Africa, but this time Ethiopia, to about 190,000 years ago, and that's the one that you see in that picture there. There are some others that appear to be kind of intermediate, also from Ethiopia, dated to around 160,000 years ago. So this was a process. It's not like at one point in time, boom, Heidelbergensis, just snapped its fingers and became Homo sapiens. It might have been a process that was ongoing in different places with different timelines, and then eventually through gene flow, just kind of merged and became anatomically modern Homo sapiens. So most of the kinds of archaeological, if you could say, or material signs of what we would consider modern human behavior had already appeared in Africa between 250,000 years ago and 60,000 years ago. So, for example, the stone tool technology, of course, kept improving. Neanderthals had very sophisticated technology, but early modern Homo sapiens were able to make these long, thin blades, which are actually very difficult to make. Requires quite a bit of skill. The earliest ones have been dated to somewhere between 280 and 240,000 years ago from Kenya. Here are some from South Africa around 70,000 years ago. So you see the symmetry and just kind of like how sharp and also like fragile. It's difficult to make stone behave in a way that allows you to create that kind of a shape without having real skill in doing that. It's not an easy thing to do. Sorry about these animations. I need to fix a little bit of that. But here's another example of some types of stone tools that we would see. These are called microliths, this sort of grouping of stone tools. They're much smaller, but they can be used for very specific things. Like these may have been embedded in spears, like you see in the picture there. This example is from South Africa, also between 72 and 60,000 years ago. Here's a bone point that was found in the Democratic Republic of Congo that it's got a pretty wide date range. It's not dated very specifically, but still old, no matter how you look at it, between 174,000 years ago and 82,000 years ago. Again, made out of bone, probably used as some kind of a like a harpoon or something for like spear fishing. And then we have sort of symbolic behavior, so something that you might consider art. So we have these examples of ostrich egg shells that have been decorated with geometric patterns. So geometric patterns are kind of like the first things that you see in terms of symbolic representations in art. And, you know, they don't serve a particular functional purpose necessarily, but they represent something. You know, it might not just be for aesthetics. It might be, you know, because they were thought to have some powers, or it could just be representative. You know, it could just be aesthetic. But these are from about 60,000 years ago. There's also jewelry, again, like these shells that have been drilled through, made into a necklace, painted with that red ochre pigment. So these are from Morocco around 82,000 years ago. And this is actually a chunk of ochre that has been, again, decorated with this geometric pattern. So that pattern is not just from like shaving some off so you can sprinkle it on something. That's been carved in there, almost doodled, like a graffiti. From 77,000 years ago. So as far as like when modern Homo sapiens started to migrate beyond Africa, we'll take a look at first the fossil evidence, and then we'll look at some genetic evidence for an independent line of evidence to sort of, you know, either support or refute the fossil evidence. So the earliest evidence that we found that's quite puzzling is from Southeast China. It was some dentition might have come from I'm not sure if it came from a mandible or if they just found the dentition, but it's dated. The dating is kind of uncertain. You know, so this is a controversial thing. Because of the context it was found in, I think it's hard to date it specifically, but somewhere between 80,000 years ago and 120,000 years ago, supposedly. But this is kind of spurious. It doesn't really fit with the rest of the data. So the other earliest thing we have is in Laos from 60,000 to 46,000 years ago. So it's a mandible. We have a skull in Indonesia between 46 and 34,000 years ago. Then we see a mandible in China around the same time, 42,000 to 39,000 years ago. And we have evidence of humans in Australia by, you know, around 40,000 years ago. But I think there's even earlier evidence beyond fossils that would lead us to believe that people were there by at least 50,000 years ago. And some scientists, even though this isn't the majority opinion, would push that back even further. So this first route out of Africa followed a southern sort of coastal route, and I'll explain that more in a later slide. The earliest thing that we see in Europe from modern Homo sapiens is around 40,000 years ago, a skull in Romania. So even though it's like geographically closer to kind of the homeland in Eastern Africa, people didn't go that northerly route until later. So as far as the genetic evidence, let's kind of back up and do a quick review. So we know there's the main type of DNA that helps your cells to function and construct your cells, basically your body plan and everything that goes on in it, comes from nuclear DNA, which I don't know if you can see my cursor here, but I'm circling the sort of eggplant type of thing in the middle, the spherical thing in the middle, the nucleus that contains the chromatin, which is DNA that's wound up, right? And that's the stuff that comes together to become chromosomes during mitosis and meiosis, right? The problem with nuclear DNA, so there's a karyotype of all the different chromosomes. The problem with nuclear DNA, as far as using nuclear DNA to trace human migrations over time, is recombination. So in order to trace migrations over time, you want to be able to kind of see a sequence of mutations that occurs in a specific chunk of DNA and look at the people who have those mutations and where they're found in the world and kind of work backwards from that. But recombination makes it tricky because let's look at chromosome 1. You've got one of those from your mom, one of those from your dad, right? When you're making your gametes, sperm or eggs, those things, they mix and match, they recombine, and in one of your eggs or one of your sperm, what you have is this hybrid chromosome 1, right? That's got pieces from your mom's, pieces from your dad's, and you pass on a shuffled mix of that. Then when you have a partner and, you have kids, if that's in the cards, it would become even more hybrid and sort of Frankenstein-y. Not that that's a bad thing. So following one bit of DNA through time that way becomes very difficult because that piece of DNA becomes smaller and smaller and smaller, and it's not always passed on. It's random which chunks get passed on and which don't. So voila. So for those reasons, mitochondrial DNA and Y chromosome DNA, which generally don't recombine and are passed in a very linear fashion, are better for tracking human migrations. So mitochondrial DNA comes from these kidney bean shaped things here in the cytoplasm, the mitochondria. They have their own DNA that's separate from your nuclear DNA. And that DNA only gets passed on from the mother, right? So the mother will pass that on to her children, daughters and sons, but only the females can pass it on, right? So the bottom on this is you, let's say, and then above you is your whole family tree stretching back generations, right? Your nuclear DNA is a combination of all the ancestors that came before you. Of course, the further you go back, the less of the DNA you actually have inherited, right? You've got half from your mom, half from your dad. But then from each of your grandparents, you only have a quarter of their DNA, And then from each of your great-grandparents, you only have 1/8 of their DNA, right? So the further back in time you go, the less you have. For mitochondrial DNA, that only gets passed along the maternal lineage, right? Just from your mom, your mom's mom, your mom's mom's mom, your mom's 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 mom, and on, right? And that gets passed on without recombining. So if there's a novel mutation in there that happened, when your mom passed it on to you, right, then you can say, aha, that mutation didn't show up until this point in time, right? So in order to retrace these steps of migration, scientists would collect mitochondrial DNA from people in populations around the globe, right? And then they would try to make some sense out of it. They would try to see how those different sequences are related to one another because they're being inherited. So if there's a mutation in, your mom's mitochondrial DNA and that gets passed on to you, right? Then you pass that on to your children, and then if there's another mutation, they will pass on the one they inherited from you and the new one to their children. So they would kind of look at these sequences and they would try and look, what's the one that has the most in common with all the other ones, right? So this one at the top here, And then the one at the bottom has a mutation from C to G, just this one mutation, right? So that kind of, okay, sequentially that sort of follows. Then this third one, it inherited that G mutation, but it also has a T to C mutation, right? Then we've got these other two that have those first two mutations, the C to G and the T to C mutations. You can see that in those bottom two. Then each of them inherited a mutation from some sequence that we don't yet have there. So there is a C to A mutation. Okay, you see how both those ones at the bottom have that. But then they each have their own novel mutation. One has a T to G mutation, and the other one has a G to C mutation. Okay, so they might label this, okay, sequence 1, sequence 2, sequence 3, and then sequence 4A and 4B. There's some mystery sequence, sequence 4, that probably just hadn't been sampled yet. This is hypothetical, like I'm not pulling this from a real case, but this type of thing happens, right? You're just sampling, you're not getting everyone's mitochondrial DNA. So this is the inferred progression, right? Sequence 1 gave rise to sequence 2, which gave rise to sequence 3. And then there's probably some sequence 4. And then there was a split where some folks who inherited sequence 4, some of those that had that split off in one direction where 4A evolved, and some of them split off in another direction where 4B came about. So to figure out where in the world people moved and in what sequence they moved, they would look at this mitochondrial DNA and they would say, the place where the highest frequency of a particular sequence exists is most likely the place where that sequence first arose, right? So sequence 1, oh look, it's in, northeastern Africa. And then it's at lower frequencies in the Arabian Peninsula, the Levant, and even lower in the Caucasus and very low in Europe, right? Sequence 2 is at the highest frequency in the Arabian Peninsula and the Levant. And there's a little bit in the Caucasus and Europe. Sequence 3 is at the highest in the Caucasus, right? And then sequence 4A is found mostly in Europe, and sequence 4B mostly in Central Asia, right? So they would infer that migration pattern looked something like this, and at some point sort of split. Some section of the population went this way, some section went that way. And this is over long periods of time, right? The Y chromosome works the same way, except it's passed on from father to son, father to son. Now, when scientists have actually done this, they have found that both the kind of earliest, if you were to trace everybody back as far as you could, the first mitochondrial DNA sequence that everybody can trace to is found in Africa. And the same thing for Y chromosome DNA, which backs up this out of Africa hypothesis, right? These are sometimes called mitochondrial Eve and Y chromosome Adam, or scientific Eve and scientific Adam. But that's sort of a misnomer because they're not the first humans that existed. They weren't the only male and the only female that were alive at the time, and they were not husband and wife. They lived at different times. That just maybe I'll explain it a little bit more in another slide here. But they can estimate that time by assuming that mutations happen on average kind of at regular intervals. Every so many generations or every so many years, there's another mutation that is passed on in that lineage, right? So they count the number of differences between the sort of original sequence and whichever one they're looking at, and that will tell them, how much time has had elapsed between those two. To understand this mitochondrial Eve thing, here's like a little schematic. So at the bottom, let's say that's all the people alive today. The dark blue individuals are women who are carrying this like sequences today, mitochondrial sequences that can be traced back to this mitochondrial Eve, right? Now you'll notice mitochondrial Eve is that dark blue woman in the top line. You'll notice she's not the only woman that's alive. She's not the only man that's alive. She's not the only person that's alive. The reason that we can all trace back to her in terms of mitochondrial DNA and not any of the other women that were alive at that point is because those branches died out in a way. That doesn't mean all the people like were killed in some kind of, horrible natural disaster. Think of it this way. Like in my family, I'm one of 4 boys. Okay. Now we all have my mom's mitochondrial DNA, but we can't pass it on, right? Our wives will pass it on to our children if such a thing happens. So even though like I am going to pass on genes I inherited from my mom, I won't pass on her mitochondrial DNA because that only passes through the female line. So, that will happen just randomly enough times, right? And so a lot of these lineages will die out, right? And or maybe in a family there's one daughter and that daughter dies early, or that daughter doesn't have children, and so she doesn't pass that on. But at some point we have to connect to someone, right? And so this is the first person that everybody alive today can trace their mitochondrial DNA to. And she lived in Eastern Africa around 200,000 years ago. Ta-da, there she is. Finally, another thing that we can look at that supports this idea of a migration of modern Homo sapiens out of Africa is that genetic diversity decreases with distance from Africa. So if you measure populations around the world and you quantify kind of the amount of genetic diversity within that population, you find that there's a correlation with distance from Africa. So these orange dots here are all within the continent of Africa, right? The light blue ones are in the Middle East. The dark blue ones are in Europe. The green ones are in Asia. And then Oceania, so Australia, New Zealand, Melanesia, Papua New Guinea. And here you have indigenous American populations. So the further you go from Africa, the less genetic diversity you have. And that has to do with founder effect. If you remember the giant bag of M&Ms, taking out a handful of those, and you're just going to have like maybe not a complete representation of the amounts of each color that were in the original bag, right? So to kind of summarize, the climate at the time that people were leaving Africa was, colder, drier than it is today. So you see there's large swaths of Africa, the Middle East, Central Asia, even Northeastern Asia that are really quite dry and deserty. And there's this little oasis kind of in East Africa, which is probably where, the population of modern Homo sapiens that populated the world really came from. And the thought is that they probably crossed, we don't know for sure, they probably crossed the Arabian Peninsula, actually across the Red Sea. They could maybe see the other side, actually, from the shore. It's not that far. And they probably took rafts or something like this. And people followed the coastline as they were moving because, the environment is a lot more similar to what they're used to in terms of being warm and having access to resources, right? If you're along the coast, you have lots of access to plenty of resources. So that first route out of Africa was a southern coastal route, which explains why humans reached Australia before they even reached Europe, because traveling through all this desert and colder tundra and whatnot would have been quite difficult at that time. So there might have been a small early expansion out of Africa around 120,000 years ago, which would explain some of this questionable evidence, like the teeth that were found in China, and some some not fossils, but material evidence that might indicate human existence in places like India and Australia early on. But it appears that if there was a small migration out of Africa at that time, it didn't leave a genetic signature. So all the descendants of that first wave didn't go on to populate the rest of the world. There was definitely a more major expansion around 60,000 years ago from the evidence. And as I said, that first route was the southern coastal route. It's important to note that people were not like, I hear what's nice this time of year is Australia. They didn't have a specific location in mind. They were only traveling very small distances, to follow game or because of population pressure. They did a calculation that if people even moved on average a mile a year, they could have reached Australia from Africa in 10,000 years, which is pretty small in evolutionary history. So you don't have to watch this, but this is a link to like a National Geographic documentary about collecting some mitochondrial DNA from people in Queens, New York, from all over the world, and sort of retracing some of these steps as a proof of concept. So that link is to a free video. You don't have to watch it, but if you're bored, which we all might be here in the coming days, it's pretty good. And Kevin Bacon narrates it, so that's a bonus. All right, so that's it. I'm going to wrap it up because this is long-winded, but I will be back for another lecture soon. Talk to you later.